Introduce Karolina Heyduk, who was an undergrad at University of Wisconsin. She studied economics and conservation biology. Her PhD is 2015, University of Georgia in plant biology, and then did a postdoc first at University of Georgia and then at Yale as a Donnelly postdoctoral environmental fellow. Then started here, UH 2020, in the School of Life Sciences, and is also director of the Joseph F. Rock Herbarium that we did a tour of, I believe, last year over in St. John. And her research spans genomics, evolution, and plant physiology. Originally, Dr. Heyduk was slated to give a talk on probably the unluckiest date possible, March 13th, 2020. And I'm glad that things have been rescheduled. We've learned how to do this online. And today she's talking about the evolution of complex traits, a case study in plant photosynthesis. Thank you.
Thanks, Floyd. All right, how's that look, everyone? Great. Awesome, yeah, I'm glad I finally get to do this. It was a bummer to have this canceled the first time around, but happy that it worked out now. And so the last few talks we've had in EECB have been focused on Hawaii and the Pacific, and today is a departure from that, but hopefully you all enjoy it. So most of my work focuses on the evolution of photosynthetic pathways in plants. And before I get into the nitty-gritty of photosynthesis, I thought I'd just kind of start with how I got into this topic and why I'm interested in it at all. When I was an undergrad, I did some field work with a graduate student out west. We were looking for these small little plants that are edaphic endemics growing in really terrible soils, essentially. And I looked around me and I thought, wow, these plants are amazing. They're learning to grow, or they can grow in these habitats where they're limited in so many ways, right? So desert plants are limited by nutrients, they're limited by water, definitely. And so that kind of spurred my interest in going into graduate school and thinking about how plants adapt to these harsh conditions. And specifically, the limitation of water is kind of a big one for plants. So plants, let me get my pointer set up here. Plants take up water through their roots, they move it through the shoot system and out the leaf. And water is leaving the leaf through pores on their leaves called stomata. This movement of water is really important for all plant functions, as is the movement of water out of the leaf, because as these stomata are open and water is leaving, CO2 is coming in. And CO2 is obviously sort of the basis of sugar production in plants. So those CO2 molecules are fixed by this enzyme rubisco through the Calvin-Benson cycle. And this works hunky-dory when plants have plenty of water, but as soon as they start to sense a little bit of water limitation, one of their first lines of defense is to actually shut those stomata. It's a really effective way at preventing water loss into the atmosphere. Unfortunately, it's also a really effective way at preventing CO2 from coming into the leaves. And so what happens is rubisco has used up all the CO2, and all that's left are these oxygen molecules. Rubisco is both carboxylase and oxygenase, meaning it can fix either CO2 or O2. And so in the presence of lots and lots of oxygen, rubisco starts to fix that oxygen, and then plants need to undergo this sort of costly process of photorespiration that helps plants get rid of some of those toxic byproducts of oxygen fixation. So when plants experience drought stress sort of intermittently, this is a fine process by which they can save water. But for plants that might experience drought stress frequently or for prolonged periods of time, this mechanism is rapidly going to lead to basically starvation of the plant because they can't get CO2. So CAM photosynthesis, or caracylation acid metabolism, is a modification to the photosynthetic pathway that allows plants to minimize that photorespiratory stress, particularly in the face of water limitation. So CAM plants open their stomata at night. Generally temperatures are lower and transpiration or water movement out of the leaf is a lot lower. So they're losing less water during this period. At the same time, they're bringing in CO2, and they're actually fixing it by a different enzyme known as PEP-C and converting it into a 4-carbon acid. During the day, CAM plants close their stomata for the most part, preventing water loss during the really hot part of the day. That 4-carbon acid inside the cells is now decarboxylated, and it floods the cytosol and importantly rubisco with really high concentrations of CO2, meaning that rubisco basically has no chance to ever fix oxygen, and so photorespiration is markedly decreased. At the same time, these plants are saving on water quite a bit. So here's a little bit more detailed of a view of CAM photosynthesis. Again, CO2 is coming into the leaves at night. It's converted through a variety of steps. Pep-C is an enzyme we'll come back to again throughout this talk. It's converting initially sort of an inorganic bicarbonate molecule into an organic acid. Those acids are actually stored in the plant vacuoles in the cells. And then it's during the day that malic acid is moved out of the vacuole and decarboxylated. And then the CO2 undergoes sort of the traditional C3 photosynthetic pathway for sugar production. CAM plants are dispersed across flowering plants. So it's not just one lineage that has evolved CAM, it's evolved multiple times. About 8% of all flowering plants use CAM. It's evolved at least 100 times, we think. The tips across in yellow on the tips of this tree are where CAM has evolved. So you can see it's in diverse lineages. It's in things you might expect given what I just told you, things like euphorbs and cacti that live in desert regions. It's also found in sort of tropical epiphytes, orchids, bromeliads, peperomia. All of those use CAM because they are experiencing water limitation in a slightly different way. They're growing without their roots in the soil, and so they rely on atmospheric water to sustain them, which you can imagine can sometimes be quite limiting. The other thing I want to sort of go into a bit is I presented this trait as either a binary C3 or CAM situation, and it's definitely not that. But before I get into some of those intermediate phenotypes, I just want to give everybody on the same page on what the C3 and CAM phenotypes look like. So one of the traits that we measure to understand photosynthesis is gas exchange. This is literally measuring how much CO2 is coming into the leaf and how much water is moving the leaf. In all the plots I'm going to show, it's going to be CO2. So in a C3 plant, CO2 is taken up by the leaf during the day, and then nothing happens at night. Remember, mostly C3 plants have their stomata closed at night. CAM plants, on the other hand, have high levels of CO2 fixation at night. We can also measure the amount of malic acid that these plants have in their cells. Remember, this is how CAM plants are storing that CO2. So a C3 plant should not have any day-night variation in the amount of malic acid, but CAM plants should. So at night, they accumulate the malic acid, and then during the day, as it's decarboxylated, malic acid levels drop. And so in between these two extremes are a variety of sort of intermediate phenotypes that we generally call C3 plus CAM. So a C3 plus CAM plant might have some level of daytime CO2 fixation through the C3 pathway, and then maybe a little bit of CAM function at night, and sort of correspondingly low levels of acid fluctuation. Another type of intermediate or C3 plus CAM species is called facultative CAM. These plants, when they're well watered, will look like a C3 plant, but when you drought stress them, they can upregulate the CAM pathway, both CO2 fixation and acidity. And then upon rewatering, they downregulate it and switch back to C3. So for CAM to function properly in plants, there are some anatomical requirements that we think are important for CAM function. The first one is packing of cells. So in a C3 plant, CO2 diffusion through the leaf layer to reach the deeper mesophyll cells is super important so that those cell layers can actually photosynthesize. For CAM plants, though, remember they're essentially concentrating a ton of carbon in their cells. And as they do that, there's the potential for CO2 leakage, meaning CO2 can move out of the cell and into the airspace between the cells and escape back into the atmosphere. So it's thought that CAM plants actually pack in their cells really densely to sort of mitigate that loss. If CO2 leaves one cell, it just goes into the neighboring cell. The other trait that we think is important for CAM is larger cells occupied by a really large vacuole. So that vacuole is where all that malic acid is being stored. And so the more you can store, the sort of hardier CAM you can use. So what does this look like in practice? Well, here's a C3 leaf cross-section. You can see the cells, there's sort of these air spaces in between the cells that you can see pretty clearly. In contrast, here's a CAM leaf cross-section of the exact same magnification. You can pretty clearly see the cells are a lot bigger and they're a lot sort of more densely packed into that leaf. So how does that sort of evolve in those intermediate phenotypes that I've already been talking about? Well, it turns out we don't think that that CAM anatomy is super important until a plant becomes fully CAM. So as plants sort of transition from C3 to C3 plus CAM, they can do quite a bit of CAM photosynthesis without necessarily investing in that CAM anatomy. Until eventually there's some tipping point that we don't clearly know where CAM plants, they're acquiring a certain proportion of their CO2 through the CAM pathway, and then they sort of shift to this full CAM anatomy. The other kind of interesting thing about CAM is that nothing in the pathway is new. So none of the genes that are in this diagram on the left that I've showed you before are unique to CAM plants. In fact, all of these genes are found in all plants in general. So these are 3 genes that are really critical for that initial conversion of CO2 to an acid, but all 3 of those are also found in C3 plants. So C3 photosynthesis is diagrammed on the right over here, but in C3 plants, those same genes are actually providing the TCA cycle with intermediates. So all of these genes are already functional and essentially actually performing the same biochemical processes in all plants. And the final thing I kind of want to talk about is how we probably are overestimating the complexity of CAM based on how, as a community, CAM people have been sampling and doing comparative studies. So let's say we have this phylogeny where we have a bunch of extant tips, some extinct tips here with the cross. And let's say we have a trait that evolved here at this blue dot. Maybe it's CAM. And so that blue dot indicates where CAM evolved, and the yellow dots are all these traits that have evolved subsequent to CAM. And all the black dots are traits that are sort of unrelated to CAM at all. What we would ideally want to do is sample all of the tips on this tree and have a really fine understanding of where these traits evolve in relation to CAM photosynthesis. What happens in sort of the real world, because of financial limitations or sampling, is that we often will use a subset of species. And so we'll take 2 species, let's say A and B, that have our trait of interest, and compare them to 2 species, S and U, that don't have our trait of interest, and look at the differences between those. And what happens is that we basically overinflate the number of changes that we think are related to CAM, where really we know those yellow dots were unrelated to CAM and actually evolved afterwards. So the key here is that we've been, ideally what we were doing is we would find closer related taxa that have C3 and CAM to better understand how these traits evolve. So my work, as Floyd kind of mentioned in his intro, it spans a lot of different fields, physiology, genomics, and sort of evolutionary biology. Today, I'm going to tell 3 sort of in-progress stories. The first about anatomy, how anatomy evolves in CAM species. The second one on some early data in comparative genomics that we're doing to understand CAM evolution. And the third one sort of delving into this yucca model system that I work on. So we'll start with the anatomy, and I'll start actually by telling you what plants I even work on. I focus my research on the subfamily Agavoideae, which is in the family Asparagaceae. These are monocots. You are probably familiar with things like agaves and yuccas. Those belong to this subfamily. This subfamily also, importantly, though, has species like hosta and camassia that actually are sort of mesic forest species, or in the case of camassia, sort of wet meadows. And so this sets up a really nice comparison system where there's some species that are not desert adapted, and then some species that are sort of these iconic desert giants. So as part of my dissertation, which feels like 300 years ago, we did a phylogeny of this subfamily and estimated sort of where CAM evolved. This is a really super simplified phylogeny at the generic level. And all I wanted to show you on this is that what we found was that there are 3 origins of CAM indicated by yellow circles here. So 1 here in agave centullado, 1 in yucca, and 1 in hesperalo. There's also potentially maybe a loss in polyanthes of CAM. And so this repeated origin of CAM in this system is really exciting as a way to study how it evolves repeatedly. The problem is that I estimated how these species are using, or what photosynthetic pathway these species are using using carbon isotope ratios. So there are 2 isotopes of carbon, C12 and C13. Rubisco, it turns out, really dislikes that heavier isotope. And so if you look at plants that are using C3 and Rubisco to fix carbon, their carbon isotope ratio is really, really different from the atmosphere because they're actively getting rid of that heavier isotope. It turns out PEP-C, which is the enzyme that CAM plants use, doesn't care as much. And so their carbon isotope ratio is shifted compared to C3 plants. And so this is sort of what it looks like. It's a really nice binomial distribution where you have C3 more negative than CAM plants. But the trouble is, remember, I talked kind of at length about these intermediate species that are using both C3 and CAM, and essentially you can't tell them apart using this method at all. So they fall into this middle ground here between the two peaks, and they're basically impossible to distinguish using carbon isotopes. So to get a better feel of what these plants are actually doing, we did extensive physiology. I'm going to show you a few plots of these like this. So the one on the left will always be CO2 uptake during the day here in white and at night here in gray. And then the plot on the right will be leaf acidity measurements. So how much acid did that plant accumulate at night, essentially. And we did this under well-watered and drought conditions for most of the species I'm going to show you. So Polyanthes, the blue dots here under well-watered looks very C3-like. You can see as you drought stress it, it actually increases the amount of CO2 uptake it has at night. And it's really clearly seen by the increase in leaf acidity at night under drought. So we might call Polyanthes actually a facultative CAM plant. Manfreda is just kind of a clean CAM species. It has high levels of acidity accumulation under both well-watered and drought stress conditions, and has nighttime CO2 uptake under both conditions as well. Yucca is an interesting group because about half of the species use CAM and half use C3. So this is an example of a C3 one. There's nothing really happening under well-watered or drought stress. Yucca allifolia is an example of a CAM species, really nice CO2 uptake at night as well. Here's another CAM species, Hesperallo. This is the third and final origin of CAM in the group. And then Hosta is sort of our clean C3 outgroup. We didn't drought stress it for fear of killing it, but we have no reason to expect that it's not C3 through and through. Okay, so using this sort of better understanding of how where CAM is in this group, I've been interested in understanding how the anatomy is sort of put together through evolutionary time. At what point do these plants evolve the CAM anatomy relative to CAM itself? So again, some of the traits that we're interested in include cell size and the airspace, and then the overall thickness as well. So I actually did this once, again, as part of my dissertation. I sampled a bunch of leaves from sort of a random smattering of species. And my conclusion at the time was, well, sort of no matter what photosynthetic pathway this plant is doing, in the Agavoideae, they're all kind of these CAM succulent leaves. But the problem with my initial analysis was that sampling was pretty sparse. I chose species without really sampling all of the diversity, and also we didn't have quite as clear of an understanding of the photosynthetic pathways these plants are using. So as part of my postdoc at Yale, I went out to the Desert Botanical Garden with Ian Gilman, who was a graduate student in Erica Edwards' lab. And we resampled Agavoideae. He was sampling Cactaceae. I don't envy trying to get a piece of the stem out of a saguaro, but we resampled a bunch of these species, got these beautiful cross sections back, and used a fancy scanning microscope at Yale to generate these beautiful images of the whole leaf cross section. And so I've had a student working on this this summer, but basically there's sort of two hypotheses I might have going into this. Let's say that we see that all of these species that I've sampled anatomy for have this sort of CAM-like anatomy. They have larger cells, they have smaller airspace. We might see, if we were doing correlations of traits across the phylogeny, that there would be no correlation between a certain trait and photosynthetic pathway, because basically all of these species would have that trait. In this case, we would infer that maybe there was an ancestral origin of this CAM anatomy. Alternatively, there might be multiple shifts to CAM-like anatomy, sort of every time CAM evolves. And this would result in a really strong correlation between traits and CAM photosynthesis. So the first thing that I even noticed, remember, again, I'm trying to understand where sort of the CAM anatomy evolved relative to CAM itself. And so these are leaf cross-section examples from each of the three lineages that independently evolved CAM. And the first thing we've noticed is that they look really different. Just even looking at them, not even quantitatively, they look very different. Not only in leaf shape, but also, and it's not as obvious on these, but the orientation of vascular tissue is different between these. The differentiation between the smaller mesophyll cells and the larger water storage cells can be quite variable as well. So this already is making me think that there are probably, at the very least, along the tips as CAM evolves, sort of these fine-tuning events that shape leaf anatomy differently in these independent origins of CAM. And so this is ongoing work. This is, I mentioned a student has been working in my lab here, an undergrad, on understanding the correlation of these traits. And so this plot is showing airspace with lower, more CAM-like values in the orange color. And you can see essentially that there's a lot of shifting happening at the tips. They are correlated, airspace and CAM is correlated across this phylogeny, but a lot of it is happening at the tips. And another thing I'll note is that the ancestral value sort of inferred back here in this tree is actually quite low for C3 species. So the result that there are shifts at the tips doesn't necessarily rule out the possibility that there was ancestrally a sort of downsizing of airspace in the ancestor of all of the Agavoideae. But we need more outgroups, and so we're currently working on that. I'm going to switch gears now to looking at comparative genomics and how it relates to CAM. And so this might seem like a weird system to be doing genomics in. It's not Arabidopsis or maize, but over the last few years since I started my PhD, especially, there's been sort of this growth in genomic resources across this group. So folks are developing resources in the agave sensulado clade. I'm particularly focusing my efforts in yucca, and then other collaborators are working on hosta and other species across the phylogeny. And so yucca is a kind of a great system for this, this work. This is the genus that I would focus on. When I was in Georgia, at least, it was very accessible and clonal. The species I worked on grew along the southeastern United States coastline. It has a genome about the size of maize, so it's not unrealistic. I mentioned this already, but the important thing and kind of the cool thing about this system is that they're contrasting photosynthetic pathways. So there is a species, one of the species I focus on uses C3 here in blue, and the other species uses CAM here in yellow under, and you can see the plots under well-watered and drought stress. And then the final sort of interesting part about the system that I think is really powerful is that there's a hybrid species that is C3 plus CAM. And I'll come back to this in sort of the third part of the talk. So with the help of a JGI Community Science Program Award, they have sequenced the genomes for the two yucca species, the parental ones I just mentioned, Yucca allifolia and Yucca filamentosa. For Yucca allifolia, we have this really beautiful chromosome scale assembly. You can see really nicely the bimodal karyotype in this group. So it has 5 really large chromosomes and 25 smaller chromosomes. That's true for all of the Agavoideae. And so this chromosome assembly was a mix of PacBio and Illumina data, along with some high-C to polish. And it's really great. It's really exciting. And the C3 parent is sort of on the way. So what do we want to do with this genome? Well, lots of things, but for today, I'm just going to focus on this one gene that I've mentioned already, PepC. So this is sort of the first key enzyme that needs to function at a greater rate in CAM plants than C3 plants. Remember, it's in C3 plants, but the timing of expression and the magnitude of its expression is going to be really different in CAM plants. PepC is also kind of interesting from an evolutionary perspective. It's a gene family that has 2 main clades, PepC1 and PepC2. PepC1 is the sort of the family that's used in the TCA cycle reactions. All the C3 plants that are using TCA and the CAM plants that are using this in the TCA cycle are using PepC1. And every time that C4 or CAM has evolved that we know about, one of these genes is recruited. So a gene from the PepC1 subfamily is always recruited for CAM and C4. PepC2 is kind of a weird gene. We don't really know what it does. It's present in pretty low copy in most plant genomes, but there's no known phenotype for an Arabidopsis knockout, for example. So its function is kind of a mystery, but we know that it remains in the plant genomes. So as we were doing all the physiology that I showed you in the first part of this talk, we also sampled leaves for RNA sequencing. And so we did this in a time course manner, just like we did gas exchange, so we know what's happening over the day-night cycle. And so again, well-watered conditions, drought conditions, and then multiple time points during the day and at night. And so here's a plot of expression for PEPC1 in agave. Remember, this is a cam plant. PEPC typically has actually higher expression in the day, and then it sort of drops off at night. And so you can see that really clearly here in those two plots. Polyanthes is the one that maybe had a little bit of an upregulation of CAM under drought stress. You can see that really nicely here. The droughted plot has an increase in the expression of PEPC copies sort of in the afternoon and into the night period. Yucca alifolia is that CAM yucca species I work on, pretty clear CAM expression pattern. Filamentosa has low to very nominal levels of expression of these genes. Hesperellus, again, that other CAM lineage, still has CAM-like expression, and then nothing happening in hosta. So none of this was surprising. It was basically what we expected all along. The surprising thing was we also looked at PEPC2 expression. And so in agave, we didn't see anything. Again, that's expected. Nothing happening in polyanthes. But in yucca alifolia, it's very, very highly expressed. And it's not only highly expressed, but it's expressed sort of right before the onset of the night period, like we would expect a PEPC gene to be expressed if it's being used for CAM. And so we didn't find that in any of the other agavoides species, but we're really, really interested in why this had such high expression in this one CAM lineage. Because we have a genome, we look to see where in the genome these gene copies are. There are 4 copies of PEPC2 in yucca alifolia. And the interesting thing is that they're all right next to each other. It looks like it's a tandem duplication event that has basically thrown a bunch of copies of this gene all in a row in the genome. And so whether or not this is related to its ability to sort of recruit this gene into the CAM pathway is unknown and still sort of an ongoing part of our work. The other kind of interesting thing about PEPC2 in general in the agavoides is that it has a different sort of molecular evolution signature to it than other species. So in this blue box are all the species that are in the agavoides, so the group of plants I study. This region that's I'm kind of circling right here is thought to be important for PEPC function, essentially. There's a modification that happens to the protein and it's thought to happen in this region. As you can see in a lot of other species of plants, this region is not very well conserved, but it's really, really highly conserved without many indels or mutations in the agavoides. So it seems like there may have been some sort of ancestral retention of this gene, keeping it conserved, and then maybe that also has contributed to its ability to be recruited for CAM in yucca alifolia. But we're doing some proper molecular evolution analyses to really figure that out. So some ongoing work that we're doing with the genomic side of things is I recently got a ficus proposal awarded where they will be, again, JGI and another national lab at EMSL will be evaluating metabolomics and proteomics in the C3 and CAM yucca species, sort of towards 2 ends. We also know from preliminary data that there's a lot of differences in sugar breakdown and metabolism between the C3 and CAM species. We want to look to see how photorespiratory products differ between the 2 species. And the kind of thing I glossed over and pretended like didn't exist is that the C3 yucca filamentosa actually does have a little bit of expression of that PEPC2 gene. It's very, very low, but it is increased under drought stress, and it does have that sort of peak in expression right before the onset of the night period. So hopefully with some proteomics, we can see, is this actually converted, translated into protein, or is this just sort of nonsense expression that doesn't result in anything functional? Okay, so the final piece that I'll talk about is continuing on with the yucca system. And this goes back to the hybrid that I mentioned earlier. During my graduate work and postdoc at UGA, I went out and collected a whole bunch of accessions of all 3 species, particularly focused on Yucca gloriosa, the hybrid species, which I knew had intermediate levels of photosynthetic pathway use. So again, blue here is the C3 species, yellow is the CAM species. You can see under well watered, the teal Yucca gloriosa hybrid has pretty high levels of daytime CO2 uptake through the C3 pathway and a little bit of CAM happening at the same time. Under drought stress, it cuts off that daytime CO2 uptake and relies solely on low-level CAM photosynthesis. And so we were interested in using this hybrid to understand sort of 2 aspects of CAM evolution. The first is the anatomical requirements. So if you put together a C3 leaf and a CAM leaf genomically into a single individual, what happens? And can some of these traits that we think are really closely tied to each other actually be broken? So if the traits are really correlated in the hybrid, let's say large cells and thick leaves, we might think that there's actually some kind of genetic constraint or linkage that keeps those traits together. Whereas if there's no correlation, then those traits are uncorrelated. And we can use this variation to see either way how it relates to CAM function, if at all. The other part that we're interested in using this hybrid for is to understand the effect of a hybridization on expression of CAM. So these hybrids we know are not all F1s. This is a hybrid index plot with one parent down here at 0, the other parent up here at 1. If these were all F1s, we would expect them to be on this 50.5 line, but we see that they're not. This is based on SSRs, but we've recapitulated this using sort of whole genome SNP data as well. So we know there's genetic variation in these hybrids, and so the question is there also physiological and anatomical and gene expression variation in the hybrids? So we did a whole bunch of physiology, lichoric acid exchange, titratable acidity measurements, at the same time collecting hundreds, almost 1,000 RNA-seq libraries. This is the plot of their sort of PCA of their expression. The lighter colors are the daytime colors, the darker colors are nighttime colors. So it's kind of nice to see that they're at least separating out correctly by time. And so the first thing I'll show you is sort of the variation within the hybrid for some of the physiology. The plot I am showing here, drought, total CO2 assimilation or uptake at night under drought stress on the Y and under well water conditions on the X. And so anything in this right-hand quadrant has net positive nighttime CO2 uptake under both drought and well water conditions. So if we plot the species, we can start down here with the C3 species. It's 0 and 0. That's what you'd expect. There's nothing happening at night. Conversely, the CAM species has high levels of nighttime CO2 uptake under both conditions. The hybrid is here in gray. These points represent individual genotypes with replicates. And so you can see they have significant, or they have non-zero CO2 uptake under both conditions. And the other thing that's interesting is that some genotypes have higher levels of CO2 uptake under well water conditions, and others have higher levels of CO2 uptake under drought conditions, suggesting that some of these can upregulate CAM under drought and some of these cannot. So we looked at anatomy as well in the three species, and particularly across genotypes of the hybrid. These are now individual plants and not genotype replicates. But this is correlating nighttime CO2 uptake sort of in sum or in aggregate, and then airspace between the cells. And so you can see the C3 plant has really high airspace between the cells and no CO2 uptake at night compared to the CAM on the other end of that spectrum. If you were to correlate across all of these three species, it's a significant correlation between airspace and the ability to take up CO2 at night. But there's no correlation within just the hybrid individuals, suggesting that airspace doesn't really seem to predict the ability of individual hybrid plants to be able to use the CAM pathway. Similarly, we can do the same kind of thing for cell area. So CAM species has really large cells, can use CO2 uptake at night. C3 plant is on the other side. Again, this is correlated across species. And again, it's not correlated within a species. So there's no correlation of cell area to nighttime CO2 uptake or CAM ability in the hybrid individuals. We can also look at gene expression across these genotypes. So now every plot is a different genotype. The coloration is a little bit different, but again, we have daytime samples and nighttime samples, and well-watered in blue and drought in red. And so this is an expression plot of PEP-C. You can see it's kind of high in a lot of these species. There's not a lot of variation happening. Maybe something interesting happening in 19 where there seems to be an upregulation under drought stress. But PEP-C is actually only part of the story. So PEP-C is taking that bicarbonate. Eventually, it downstream gets converted into malate. The interesting thing is that malate is actually a negative feedback regulator of PEP-C. So as malate accumulates, PEP-C decreases in its ability to function, with the exception of when it's phosphorylated. So PEP-CK is a kinase that is dedicated to the phosphorylation of PEP-C. When PEP-C has been phosphorylated, it's no longer sensitive to malate, and it can continue chugging along this pathway and producing more and more malate. So this phosphorylation of PEP-C is really, really important for CAM plants to be able to function in sort of their high malate cellular environment that they're producing. So we can look at PEP-C expression. Again, a lot of these look the same, but again, 19 jumps out as something really different happening, where there's a really strong increase in expression of PEP-C-K, this is the kinase, at night. And so we have many more, we have 20 some genotypes that are sequenced with RNA-seq, so we have many more comparisons to do. But it looks like ideally we will be able to use the variation in CAM and expression to sort of tease apart which genes are contributing in different genotypes to the CAM pathway. The final thing I want to tell you about is we've also been looking at total repetitive element content in plants, or in these plants. So plants in general have a lot of repetitive elements in their genomes. They can be really important for novel function, including silencing or duplicating genes. And so we looked at total transposable elements in the genomes of the CAM species, Yucca allifolia, the C3 species, and then the hybrid. And so there are differences sort of genome-wide. There are also differences if you look at individual plants, or individual genotypes, excuse me. This is across multiple different classes of repeats. You can see there's definitely differences between the species, but also within a species, there's some variation. And that variation can be seen really clearly in some of these plots. So for example, gypsy elements, which is a whole superfamily of repetitive elements. There's not a ton of difference between the three species, and in fact, gloriosa kind of looks just intermediate. Other elements have sort of this transgressive phenotype in the hybrid, where there's actually more of this repetitive element in this hybrid than either of the two parents. This is the sort of pattern you would expect to see if there's some genomic shock happening after a hybridization event, where suddenly, because those two genomes are combined in a single individual, they're allowed to sort of proliferate in the genome. And then others have a sort of a decrease in abundance in the hybrid relative to the other two parents. So ongoing work in this realm is also using the ficus proposal I mentioned earlier, again, looking at metabolites and proteins, specifically in the hybrid, and specifically comparing genotypes that can upregulate CAM and those that cannot. So sort of understanding what signaling pathways are present or not present in some genotypes that allow them to turn on CAM under drought stress. And we're also interested in sort of looking at the parental gene source in the hybrids to understand if that's related to CAM function, as well as TE or repetitive element locations in the CAM or in the hybrid genotype genomes to understand if those at all are affecting CAM expression in those genotypes. All right, so that's all I got. Lots of people to thank both at UGA and at Yale, and the JGI Hudson Alpha folks have been super important in helping me generate really awesome genomic resources. So hopefully you enjoyed a little trip into desert habitats and physiology, and I'd be happy to hang out and take some questions.
So I have a question, if I can jump in. Can you artificially hybridize these two species, and then are the hybrids different from the wild hybrids that have had a chance to adapt?
Yeah, so I glossed over this because I was trying to convince you all this is a great system, but the one really downside to studying yucca is that they take a really long time to flower, up to like a decade. And then I have not been able to create a hybrid artificially. I have tried, and something terrible happened every time. One time I pollinated a bunch of flowers and they all got like buzz-sawed off. So I haven't been able to recreate the hybrid personally. I know some folks in, I think some European botanical gardens have created the hybrids, and as far as I can tell, they look the same, but there hasn't been like any extensive study on them or their functionality.
Hi, I just want to ask you a question, Karolina.
Hey, Mark.
Comparison, hi, you worked on these yucca species on the eastern coast of North America. What about on the western coast? There's yucca whippley, also now I think known as Hespero yucca. Yep. Yucca. Is that species and that genus now, how does that fit in, Hespero yucca, and is that whippley, which is not endangered, but it's getting rarer, is that species CAM species?
Yeah, so Hesperia yucca whipplei is actually sort of in a clade that's sister to yucca and agave. So it's fairly distantly related within the Agavoideae to yuccas proper. And the consensus on whether it's doing CAM or C3 is kind of out. I had some plants and they were misbehaving in the growth chamber and I needed to redo that experiment. I think if anything, they're probably weak CAM, so able to do a little bit of CAM. Yeah.
How long do they take before they flower?
Those I don't know. I would imagine it's on the same order of 7 to 10 plus years.
Thank you.
Yeah. Rosanna.
Carolina, thank you for the presentation. It was fun. I have a question for you. I don't think I've ever asked you this, but it is a hybrid, but it's not a polyploid, like an allopolyploid, right?
Mm-mm.
Okay, so what's happening with those super conserved regions of the PEPs? Because, you know, I would sort of expect that after a polyploidy or allopolyploidy event in which, like, you know, you want to conserve this because the hybrid, right? Like the hybrid behaves well versus other things that are non-polyploid are sort of free to evolve. But it doesn't seem to be the case in your system. So
yeah, so there's a few things
of ancient polyploidies that might have produced that.
Yeah, so the hybrid itself, I didn't mention this, it's a homoploid hybrid, so there's no increase in genome size after the hybridization event. It's essentially taken half of the chromosomes from one parent and half the other and smushed them together, which is nice because then I don't have to worry about the effects of polyploidy on the CAM phenotype that I'm interested in. But Rosanna, what I think you were talking about is that alignment of PEP-C where there's that conserved area. And so that alignment actually was across all of the Agavoideae, which have had some genome increase. We're not sure if it's a duplication or a triplication early on, but almost all of the species share that initial whole genome event. And so, yeah, it's curious that, you know, even if there are multiple copies, they all seem to be retained. Yeah, there's a lot of stuff that I haven't dug into yet, but yeah, we should chat about it.
I have a question, Karolina.
Hey, Kurt.
Hey, so I was wondering, you have your two species, and can you tell a story about the common ancestor and how those two species evolved? Was it related to environmental differences, and what was the time frame for that evolution of those changes that we now see today?
Yeah, so you're talking about the yucca species that I work on a lot. Yeah, so the yucca itself is probably only about 10 million years old. It's pretty young. And the sort of the sister group to all of the yuccas are actually Joshua trees, Yucca brevifolia. Those we know are C3, and so we're pretty confident that the ancestor of yucca was probably C3. But what that looked like is a little unclear. So there are some, it's unclear for a few reasons. The first is that there are some traits that are shared between the C3 and CAM yuccas. So some of the C3 yuccas do have this sort of larger cell size and smaller airspace than might be expected for a true C3 plant. So was that ancestral, or is it actually just something about the environment that they live in that's selecting for that kind of phenotype, let's say water storage in the desert? I also mentioned that the gene expression of PEP-C was kind of similar between the CAM and the C3 yucca, although much lower expressed in the C3 species. But still, that expression pattern is not really expected in any true C3 plant. So it's been kind of one of my hypotheses that early on, the ancestral individual of CAM lineages probably had some of these traits already there, including maybe some changes to the expression of PEP-C that are related for CAM. And so the other kind of interesting thing about the CAM and the C3 yuccas, they're actually, the ones that are CAM are all in one subgenus, the Sarcocarpa, these fleshy fruited yuccas. The C3 species are all in Chenocarpa, which are these like dry fruited species. And their ranges are not distinct. They actually overlap quite a bit in the southwestern United States. And so sort of all of the easy things that you might imagine might be happening, like, oh, well, you know, all the camels just shifted into desert regions. That's not true because they actually grow right next to their C3 relatives. So it's a complicated, I don't think I told a good story, but I was just telling you all the complications of the story.
That was good. I think it's interesting that with the distributions being overlapped and still having the differences in photosynthesis.
Yeah. Yeah. Any other questions?
Okay, so thank you.
Yeah, thanks everyone.
For this. Can everybody unmute their microphones and just clap for a second to thank Karolina?
Okay with emojis.
Yeah.
All right.